I wanted to show a little bit to you some of our latest findings on what are the basic properties of TDP-43, especially those that could be connected with disease. And the reason why is that whenever I go around talking with people, I always get the question, what is TDP-43 doing? Unfortunately, I don't have the answer to this question yet. But what I'm going to try and tell you a little bit in this 40 minutes or so is what really TDP-43 does inside the cells and what are its connections. So the first thing that I think everyone should always keep in mind when you talk about TDP-43 is that we are not talking about a brain-specific protein or a protein that is expressed only in a particular developmental stage. It is an ubiquitous protein. It is expressed in all the cells of our body, although to varying extent, and also it is expressed right from birth to death of any organism. And the reason why is that TDP-43 belongs to a very old class of nuclear proteins, probably the most studied class of nuclear proteins beside the histones themselves. And already in the early 80s, right, people were aware that there was a huge number of proteins that were associated with heterogeneous RNA. And the reason why all these proteins are now known as H9MP, A, B, and C is because with the limited technical resources that were available at the time, the first way that people try to differentiate them from each other was to actually run the very simple and basic STS-PAGE gels and simply naming the proteins that were more abundant based on alphabetical order. So luckily enough, they didn't run out of letters before before finding all the members. And so now we know more than almost 30 years later, that really HNRMP proteins that are named from A to U are a class of very heterogeneous polypeptides of highly variable length, but they're all characterized by several characteristic features. And first of all, there is the presence of one or two RNA binding domains, and then there is normally the presence of several regions that are GLI-rich, proline-rich, glutamine, and asparagine-rich, and these regions are almost always involved in protein-protein interactions. And all these proteins, what these proteins do is they really act as a connection between all the different stages of an mRNA life cycle. So starting right from the transcription event, then all the other processing events that the mRNA undergoes, so the splicing, of course, but all the modifications, the transport, the translation itself, they are mostly mediated by members of the H9MP proteins. And one of the things that should be borne in mind, actually, this diagram that I did, I can criticize it immediately, because if you look at this diagram, it looks as if TDP-43 is working independently on all these processes. But this is not actually true. I don't know if you're familiar with the old poem by John Donne, right? No man is an island. And this applies very much to HNRMPs as well. So no HNRMP is an island. And really, all HNRMPs are working together, talking to each other to actually fine-tune all these processes. And this is something that we now know very well, because, as Philip said, there are a lot of proteomic approaches that have been tried on TDP-43, and now we know many proteins that are that can be bound by TDP-43, and of course whose regulation and functionality can be affected by TDP-43. But already before TDP-43 became famous, what I did was, and not to underscore the property of the novel high-throughput proteomic approaches, But with a very simple experiment in 2005, what we were actually interested in were to look for the major partners which were bound by TDP-43 inside the nucleus. And so what we did was to use this very simple GST overlay assay where you actually put nuclear extract on your western blot membrane, and then you use your protein as a probe to see which of the proteins that are available, that are immobilized on the membrane, are recognized by your protein. And the result that we got is shown here, and as you can see, there is a number of proteins that were specifically recognized by TDP-43, and all these proteins belong to the basic HNRMP class made up by A1, A2B1, A3, and HNRMP-C. So, first of all, what we tried to do later on, when D'Ambrogio was young, as Tito says, was to see if there was some kind of functionality associated with this interaction. And what D'Ambro did in the lab was to set up a very nice system that we have used very proficiously since then for a number of projects, and that is the add-back system. So what we did is to actually, if you have a mini gene that carries CFTR exon 9, you can see that in normal conditions, most of the CFTR exon 9 is skipped because TDP-43 binding to the 3' splice site is acting as a negative splicing inhibitor. If you remove TDP-43, then you can actually release the inhibition and you can get a big increase in CFTR exon 9 inclusion. If you put back an SI-resistant TDP-43, you recover the inhibition. But if you take a TDP-43 that has a C-terminal truncation, and D'Ambro also in his experiment managed to narrow down the region that was bound by HNRMP-A2 and A1 in particular, and you can see that if you delete these two regions, then you cannot recover splicing inhibition as efficiently as the wild-type protein. So obviously, the interaction of TDP-43 with HNRMP is functionally important. It's also functionally important, as Tito described very much better in his talk this morning, because if you actually take the region that is responsible for the interaction with the HNRMPs, that region can be exploited to trigger the aggregation of TDP-43. And the reason why this could be so is due to the fact that if you take this region without any interactor in solution and you allow enough time, then what this region does is to actually start to acquire some amyloid-like properties. So HNRMP proteins are not just important for the functionality of TDP-43 in the pre-mRNA splicing, but they're also important probably with regards to its solubility. And you can also think of other connections, important functional connections between HNRMPs and TDP-43. So for example, you could have individuals that carry mutations, independent mutations in HNRMPs like A and B, as John described this morning. Of course, you can have, and this is something that Clotilde, for example, Laget-Turin has described for TDP-43 and FAS, where different HNRMPs can bind common targets, and so they may act in parallel or synergistically to affect the processing of mRNAs that are important for the pathology. And also, in a way that you could think of, is the fact that if you have in the cell other RNA repeat expansions like C9orf72, then these other repeats could sequester HNRMPs that are bound to TDP-43 and therefore affect its functioning. And this is something that we really try to do, investigate, and we have just started a little bit better, so most of the data I'm going to show you have not been published yet, at two levels. The first level is at the level of the organism, and the second level is at the level of potentially important splicing events where TDP-43 and together with HNRMP HNRMPs could play a role. So at the level of the organism, what we decided to exploit is the model that was set up by Fabian in 2009. And the reason why we decided to exploit this model is that if you use this very basic, as I've told you, GST overlay technique, and you look at the proteins that are recognized in the nuclear extract of human and Drosophila, you will see that if these are the HNRMP proteins that are in the human extract, you can see that the human protein in the Drosophila nuclear extract is recognizing this set of proteins that are that is migrating almost in the same position, and the same also is true if you take the Drosophila protein. The Drosophila protein can actually bind all human HNRMPs and its own HNRMPs, and the human protein can do exactly the same. And so, thanks to the work of Maurizio Romano in the lab, we managed to actually identify, using co-immunoprecipitation analysis, the regions that were involved in these cross interactions, and not very much surprisingly, although the C-terminus of TDP-43 and TBPH is not that similar, the regions that were involved in these interactions have been completely conserved during evolution. And the other thing that I wanted to say is, Drosophila for us was an ideal system as an organism to test this connection, and also because, thanks to the work in Fabian's lab, we also had some endogenous targets that were known to be affected by TDP-43, and in this case, I'm showing FUCH, that is the homologue of MAP1B, that we could test for alteration. And very, very pleasingly so, what we did together with Fabian was to build Drosophila models where we didn't completely knock out the expression of TDP-43 or of HRP38, because knocking out the expression of either of these two proteins results in a completely lethal phenotype, well, not TBPH, but HRP38 for sure. And the genetic interaction between these two proteins can actually be seen very easily by looking at the lifespan of the Drosophilas, because you see that these are normal flies and controls. If you partially knock out TBPH or HRP38, that is the human homologue, the Drosophila homologue of HNRMP A1 A2, you can have a loss in the lifespan of the animals. But if you partially knock out to exactly the same level both TDP-43, TBPH, and HRP38, you can see that the lifespan is very much dramatically reduced. And this is actually connected with alterations at the level of expression of genes that are known to be targets of TDP-43. So you can see in this case, this is adult fly's brain, and you can see that there are defects in the regulation of FUCH, whether you partially remove HRP38 or you partially remove TBPH, but these defects are much more pronounced when you put these two proteins together. So this doesn't really tell you, there may be additional targets, of course, but what this is telling you is that whether they work together or independently, as far as the pathology is concerned, there is a functional relationship between the levels of TDP-43 and HNRMP proteins. And so now, thanks to the recent support of the foundation from Thierry Latran, what we are going to do now with Fabian is to start a systematic screening of all HNRMP proteins that have very good homology with their human counterpart, and to actually see which ones could be affecting the working of TDP-43 in mediating neurodegeneration. And so, just switching gears a little bit, what I wanted to tell you a little bit now is, how are TDP-43 and HNRMPs working together to affect RNA processing. And basically, you can see that HNRMPs work in many different ways to affect pre-mRNA splicing. So, like TDP-43, they could just, bind in proximity to splice sites, and also HNRMP1, of course, and all these other HNRMPs, they can bind in proximity to the splice site, and they can interfere with the binding or splicing of basic splicing factors. They can actually interact with each other to either loop out particular exons and cause them to skip, or actually to bring particular exons together and actually enhance their recognition. And there are several models that have been found and described in the literature. And the problem with applying and trying to understand what is the connection between TDP-43 and HNRMPs using some of these models is that we still don't know in detail, although we know many candidate genes whose splicing is affected by TDP-43, we still don't know in very much detail what are the mechanisms through which this occurs. And in my opinion, one of the most interesting examples where TDP-43 is known to play a role and where actually HNRMPs could contribute to this role is represented by a particular exon that we described with Leonardo Petruccelli 2 years ago, and this particular example is represented by a particular exon called exon 17B that is found in the SORT1 gene. And the SORT1 gene could be extremely important for the pathology because it has been recognized as the major receptor through which neurons can actually take progranulin inside their body. And as you know, loss of function mutations in the progranulin genes are always associated to very severe cases of frontotemporal dementia. But the peculiar issue about the regulation of TDP-43 in this exon, what we found with Leonardo, is that if you take the mouse SORT1 exon 17B and you remove TDP-43, you get a very efficient recognition of this exon. So this exon is basically a alternatively spliced exon regulated by TDP-43 that can be either included or excluded in the final mRNA, depending, of course, on the concentration of TDP-43 available inside the cell. In the case of the human exon, the situation is quite different, because if you remove TDP-43, then what happens is that the levels of inclusion compared to the mouse are very, very much reduced. And there is a functional reason for this, and this is why I think it's extremely interesting as far as the pathology is concerned, because these two exons, and together with the flanking intronic sequence, they are very, very similar. But there are inside the exon that is a 99 nucleotide long exon, there are just 8 nucleotide differences between the human and the mouse exon. But one of these differences, from the coding point of view, the protein coding point of view, is extremely important, because the substitution of the C for a T in this position here actually changes the reading frame, and this T codes for a stop codon. So if in the mouse context, inclusion of this exon 17B will give just the formation, will just give the result of making a SORT1 receptor that is functionally functionally identical to the receptor lacking exon 17B, and this is what Leonardo proved in his lab using functional tests of progranulin uptake by cells that were transfected with these two different isoforms of the receptor. In the human situation, the presence of the stop codon in exon 17B will cause the production of a toxic isoform that cannot be incorporated in the cell membrane. It will be released outside and therefore can act, for example, as a decoy to lower the amount of progranulin that can be uptaken by the neurons. So what we think that is going on is that besides just the presence of TDP-43, there are obviously other mechanisms, and mechanisms that are most likely mediated by other HNRMPs, that could make, that could actually cause the human exon to be much more heavily repressed in the human context as opposed to the mouse. And this is something that also comes from some preliminary experiments that we have done in the lab. So we have started to look with Fateme Mohagegi, that is our latest PhD students. We have started to look at what happens if we look at a more extensive panel of human and mouse cell lines using mini-genes that carry either the human or mouse exon 17B, and to see what was the effect of TDP-43, removal of TDP-43 in these different cell lines. And you can see that you get a pattern that is really consistent with the fact that TDP-43 is not the only regulator of this particular exon recognition, because, for example, in human HEP3B cells, the exon 17B is never included whether you have TDP-43 in the cells or you don't. In some cells, there is a complete switch of exon inclusion from mostly skipping to mostly inclusion when you remove TDP-43. And in some other cells also, you start to see that if you remove TDP-43 from in the human context, and if you remove TDP-43 and you look at the human context, you actually get some very nice inclusion of this particular exon. So one of the reasons why this could be, and at the time when we were studying this exon, because we were rather in a hurry, we didn't know. So at the time when we started looking at this exon, what we did was to not simply look for just the presence of TDP-43, that you can see is almost the same both in the human and mouse context, but we were also aware of the fact that other HNRMP proteins that are known to have a negative impact on splicing, and also we looked for positive enhancers of splicing regulation. So there were lots and lots of these proteins binding in the vicinity of the human and mouse exons. And so what we have done with Fateme in the last few months or so is to actually look a bit closer and not just which proteins are binding and which are not, but also where they are binding within the human and mouse context. And so we have extended the list of these proteins, and we have also looked whether these proteins are binding to the upstream intron, the human or mouse exon, and the downstream intron. And you can see that although the pattern is similar for several factors like HNRMPH or HNRMPF, or for example TDP-43 itself, that you can see can bind only to the upstream intron and, of the human and mouse, because this is where the TGs are localized, there are some others that have some quite interesting differences. So for example, we have HNRMP A1, the HNRMP A2, that is binding to the upstream intron of the human exon, to the downstream intron. The same is true also for the mouse, but the big difference between the two is that in the human exon, you have binding of HNRMP A2, but you don't see it in the mouse exon. And the same can also be seen for, there are also differences as far as PTB is concerned. But this one is particularly interesting, and it's particularly interesting because what we had done previously with Leonardo was to make a series of constructs that actually start from the human, and then on the basis of the human intronic and exonic sequence, we were actually introducing mutations, just selective changes inside the exon to actually mousify the human. And you can see, so for example, in this mutant, we just replaced this A for G. In this one, we replaced these two C's for the two T's, and so on. And we knew that these mutants had an effect on splicing, because if you put all these constructs in a mini-gene context and you look for exon 17B inclusion, you can see that two of these mutants in particular, this one and this mutant 1 and mutant 4, they were more efficient at including the SORT1 exon 17B than the wild-type construct. And when we tested for the binding of these mutants to some of these factors, especially HNRMPA2, you can see that inside the human exon you have binding of HNRMPA2, you have binding to the two mutants, 2 and 3, where you really have no difference in the splicing profile with respect to the mutant, but you don't have binding, of course, in mouse exon 17B, that is basically more efficiently included by the mutant, but the binding of HNRMPA2 to these two positions is actually almost completely abolished. And so really what happens is that I think one must be very careful when trying to interpret splicing data, and especially in trying to interpret the function of TDP-43 in any particular system, because as is shown here, and we are currently testing all the other possible candidates, but even in situations where you don't think that there is a huge amount of difference, because you look for very similar for very homologous regions, you could have differences in HNRMP recruitment in addition to the presence of TDP-43, and the presence of all these other HNRMPs can certainly have a functional effect on, can actually profoundly influence the functionality of TDP-43 itself. So just to give you the conclusions of this talk, basically what we think is that in order to better understand the changes that occur in RNA metabolism during the disease or any other type of or any other aspect of TDP-43 biology that you care to investigate, you certainly cannot just look at TDP-43 itself, but you should try and look at it in as much a global manner as possible, especially concerning the presence of HNRMPs. And also, this could actually be, at least partially, or in addition to technical differences, to an explanation of why when you compare all the different high-throughput studies that have been published on TDP-43 biology, there is and looking for genes that are affected by the presence or absence of this protein, to be honest, you find a very little overlap between all these different studies, because probably in all these cases you have to take into account that TDP-43 functionality is not just a standalone characteristic in every cell type, but probably that is profoundly affected also by the functionality of all the other interactors, and especially the other HNRMP proteins that are TDP-43 sisters in, let's say, evolution. And so I'll just stop here, and I will acknowledge the people that have worked on this project. So all the Drosophila is thanks to Giulia, Rafa, and Fabian. Then Maurizio, Cristiana, and Fateme are the ones that are working on the SORT1 and also on the Drosophila project. And then, of course, there is Tito that I've been working with since 1993, and nothing of what I've shown you would have been made without his presence. And then there is me, of course, and the people that have now started supporting the research. And this is something that, because I have time, this is something that I would like to point out. So you see, we now have support to work on TDP-43 from ADISLA, that is the new Italian agency for research on ALS. We got the support of the French Foundation Thierry Latran, and together with Peter Huting, that is here in the audience, we actually managed to get support also from the European Union, and in particular the GMP&D research project. And this is something that I think is a good lesson to take home to especially the young people here in the lab, because you must remember that in 2001, when Tito and I found TDP-43 in CFTR exon 9, nobody was giving us a single euro to do research on TDP-43. the only research on TDP-43 we managed to do is with money that was saved up from projects that at the time seemed to be, the next best thing in history. So my take-home message, to especially the young people and the people from ICGEB member states is that, the only thing that is important is that you work on something you like and you work well. Then, the future is not in your hands. You could have a goal, if you complain that the subject you're studying now may not be important for disease, TDP-43 is here to remind you that maybe now nobody realizes it, but in 10 years' time or so, you will be, I wouldn't say, showered with money to work, because money is never enough, right? But at least you don't have to write fake research programs then to work on something that people think is most important, and then save with the money that you save up, work on TDP-43. Okay, I'll stop here. Thank you.